Hi, it's Jamie Batts, your instructor for AMP2. We are continuing on with the digestive system lecture today, and we just finished talking about all of the parts of our digestive tract. Now we're going to start going into detail on the accessory organs of our digestive system. So these are organs that the food chyme / bolus, whatever you want to call it, does not pass through. Typically, these are things that are secreting into the digestive tract. So switching to screen share mode right now, and let's talk about some of these digestive organs. We have salivary glands, the gallbladder, the pancreas, the liver. We're going to go through each of these in detail. The pancreas, liver, they also have other functions other than just digestive system. So a lot of them have, I didn't realize my dog was upstairs and I heard footsteps. Sorry, freaked out for a second. Some of these organs have dual functions, not just in our digestive system. So there they are. Let's learn about them in detail. This is an overview of the digestive and metabolic functions of the liver. So here's something that I think is super important. Understanding the difference between a digestive function and a metabolic function, right? Digestion is having to do with the chemical breakdown of the food itself and the absorption. The metabolism aspect, that's having to do with enzymes and chemical reactions and the way that your body processes the nutrients that have been broken down by our digestive tract. So some of the things that our liver does is secrete bile. Synthesizing and secreting bile, that's a digestive function. Storing glycogen and lipids, that would be considered a metabolic function. That's just storing. We're not actually breaking anything down. Why are we storing glycogen and lipids? We're storing it for our metabolism later. Maintaining normal concentrations of glucose, amino acids, and fatty acids, again, that's a metabolic function, not a digestive function. Synthesizing, interconverting nutrient types, like converting carbohydrates to lipids or lipids to carbohydrates, that's a metabolic function. Synthesizing and releasing cholesterol, that is a metabolic function as well. Inactivating toxins, storing iron, storing fat-soluble vitamins, all metabolic functions. So really the main digestive function of the liver has to do with the secreting of bile. Some of the other things our liver does, secreting the plasma proteins like albumins, right? Synthesizing clotting factors like fibrinogen, phagocytizing damaged red blood cells, that's done by Kupffer cells, which are in the liver, stores blood, absorbs circulating hormones and immunoglobulins or antibodies, right? And it also inactivates and absorbs lipid-soluble drugs. You can pause the video here, go back and review this section. We're going to start talking about the salivary glands. There's 3 pairs. They secrete saliva into the oral cavity through little ducts. Each pair is slightly different, and the secretion from each pair salivary glands is slightly different as well. There is a salivary reflex, which is monitored either by the cranial nerve 5, cranial nerve 7, 9, or 10, depending on which part of the tongue or which part of the mouth where these are located. It's all done through parasympathetic stimulation. You have your sublingual salivary glands underneath the tongue, secrete into sublingual ducts on either side of the lingual frenulum. We saw them on a previous picture in another lecture. They're producing a very mucousy secretion that acts as a buffer and a lubricant, very small amount of the total saliva produced. Our submandibular salivary glands are on the inner surface of the mandible. They are secreting through the submandibular duct, and they secrete buffers, mucins, another kind of mucousy type compound, and salivary amylase. Salivary amylase is an enzyme that breaks down starches. These cells also transport IgA antibodies into the saliva, providing a minimal protection against pathogens. This is the majority of our saliva production. Our parotid salivary glands, which are kind of up back here, you see them a lot on the models in class that looks like this big blob of fat in the back. Produce a very serous-y secretion, a very liquidy secretion, and lots and lots of salivary. Amylase is just about 25% of our total saliva production, so the majority is coming from submandibular. There they are in their relative locations. Let's talk about the cells of our salivary glands. So you have duct cells, which just assist in secreting the buffers and the antibodies, the mucus cells secreting the buffers and the mucins, and then the serous cells, which secrete the amylase and the lysozyme. Lysozyme is an antibacterial enzyme. They're also helping to transport antibodies from the interstitial fluid into the saliva. There they are, the duct cells, mucus cells, serous cells. You don't have to know how to identify those on a slide. So overall, saliva is a mixture of all of the glandular secretions. We produce over 1 liter of saliva each day. The majority of our saliva is just water. The overall function is to flush the surfaces in the mouth, keep our teeth clean, maintain a relative pH of around 7 to prevent the buildup of acids by the bacteria. Also, the lysozyme and IgA antibodies there to help control bacterial populations and help mix with the food to form a bolus that is easily swallowed. You can pause the video here, go back and review this section. The next stop on our tour of accessory organs is the liver. It's the largest visceral organ, more than 3 pounds large, wrapped in a very fibrous capsule covered by the visceral peritoneum and composed of 4 lobes, the left, the right, the caudate, and the quadrate. Here you can see a nice cross-section of the body, a transverse section. So you're looking at the liver in relative position to the stomach, the greater and lesser omentum, the aorta, the spleen, inferior vena cava passes through. You can see what's called the porta hepatis, which is basically all of the vessels and how they enter our liver. So let's talk about the lobes. The left lobe and the right lobe are separated from each other by the falciform ligament. There's also a coronary ligament that is an extension of the falciform ligament. Surrounds this bare area, which is where the liver contacts the diaphragm, kind of top area. There's no peritoneal covering to this section. And the round ligament is a thickening posterior margin of the falciform ligament. It's basically marking that path of the fetal umbilical vein, which, if you recall from fetal circulation, passed through the fetal liver in order to get to the heart. The caudate lobe is on the posterior surface, as is the quadrate lobe. It's separated from the right lobe by an indentation, just by the inferior vena cava, and the quadrate lobe is near the gallbladder. So let's talk about the gallbladder, which main function is to store the bile. Bile is produced by the liver, but is stored in the gallbladder. So a lot of people don't realize that. A lot of people assume that the gallbladder makes the bile. It's the special cells in the liver that makes the bile. The common bile duct is carrying the bile from the liver to the gallbladder and to the duodenum. So it's carrying the liver or carrying the bile from both of those structures right down into the duodenum. The porta hepatis or porta hepatis, again, tomato, tomato. This is the doorway to the liver. This is that kind of hilum-like area where we have the entrance of all of these major vessels, the common bile duct, all coming and going at this main center doorway to the liver. You see that whole structure right there. You can pause the video here, go back and review this section. We're going to continue talking about liver tissue and the basic function of the liver. So liver lobules are a basic unit of liver tissue. There's 100,000 or so of these liver lobules. They're very small. They're separated from each other by a septum. Your body has ways of compartmentalizing everything, right? And the reason is to contain any disease or pathogen from getting to other areas. So these lobules are maintained by septa. They have a hexagon-shaped appearance in a cross-section, and there are 6 portal areas, 1 at each corner of the lobule. So do yourself a favor right now. Draw a hexagon on your paper. A hexagon. It looks like this, right? A hexagon. At each corner, you're going to have a portal area. So draw a circle at each of those corners. right? A circle at each of those corners. Those are the portal areas on the liver lobule. Each portal area has a triad. Get 3 different colored pencils or pens. And at each corner, make 3 dots. 1 with each color. I'm doing it just to show you. See? I don't know if you can see the color. I have a pink, I have a blue, and I have a black. I would draw 1 of those dots at each of those corners. Guess what? One of those structures, pick a color, one of those is a branch of the hepatic portal vein. That branch is receiving blood from the hepatic portal system, which is bringing the blood fresh from the intestinal tract right into the liver. Remember? All routed over to the liver first. One of those colors is a branch of the hepatic artery. And one of those branches, or one of those dots, is a branch of the bile duct. So, branches of the arteries and veins from each portal area is delivering blood to the liver. They're called sinusoids. They're basically our capillaries of our liver. So, they're delivering the blood to those areas. So, the liver lobules contain hepatocytes. These are the liver cells. These form basically like spokes of a wheel inside this portal system. And the exposed surface is covered with villi. So if you kind of, again, we're creating this. Don't look ahead in the PowerPoint, right? You have this nice little hexagon here. You're going to have a central area, so draw like a circle in the middle. And then have some spokes coming out and going to each portal. So it'll look like that. Boom. Each of those hallways is surrounded by hepatocytes. Hepatocytes are liver cells. The hepatocytes are separated by sinusoids. These are little tiny blood vessels. There's no basement membrane. They're kind of like fenestrated capillaries, but they have a larger hole. So, boom, look what we just drew, right? Here is the liver lobule. You can see exactly what we just drew right here on this image. All 3 vessels at each of the corners of our hexagon. You have these little pathways and all those little cells in there, all those little boxes are the hepatocytes, all right? The sinusoids are those pathways, the spokes, if you will, coming out from that center area. So let's talk about this anatomy and how it all works. And it should make more sense because you drew it out, and you can always redraw it and make it prettier if you like. So stick that over there. Blood enters the liver sinusoids from branches of the hepatic portal vein and hepatic artery proper. So blood's going into the sinusoids, starting out in these little corners, right? The blood's going to flow inward. About 1/3 of the blood supply is arterial. The remainder of it is coming from the hepatic portal vein, right? So the majority of that blood that's flowing through those sinusoids, those little spokes, is the blood that's coming right from the intestines. Hepatocytes that are near the sinusoids are going to regulate solute and nutrient levels by absorbing and secreting. So imagine this. You have your little thing, those little cells that are on those blue lines, and again, it's obviously, if you look back at that last slide, it's not quite as organized as this, but in my head, I like to have everything organized. I'm kind of weird. So all those little cells that are lining those spokes there, those are basically tasting the blood, for lack of a better term, dipsticking it and saying, oh, we need more calcitriol. Oh, we need more, fibrinogen. Oh, we need more albumin, or we have too much albumin, right? They're dipsticking and tasting and adjusting ion, nutrient, and enzyme concentrations. So that's what they do. You also have Kupffer cells. Kupffer cells are scattered throughout the sinusoids, throughout those little spokes. They are specialized macrophages. They're They aren't macrophages. They're just special macrophages that are in the liver. So they're there to, remember what macrophages do? Macrophages are like Pac-Man. They're there to engulf bacteria, debris, anything that doesn't belong there. They also help to store iron, lipids, heavy metals like tin or mercury that may have been absorbed from our digestive tract. The sinusoids all drain into a central vein. So that little circle in the middle, that's our central vein. Guess what? All the central veins merge together to form the hepatic vein. Guess where the hepatic vein goes? Hepatic vein goes right into the IVC, right back into the heart. Boom. Other things. The hepatocytes are secreting bile into our bloodstream. So the bile canaliculi basically merge to form bile ducts. The bile ducts basically go into the nearest portal area, and bile plays a role in the digestion of our fats. So here you see the bile canaliculi, these little canals that are being that are absorbing the bile that's produced by the hepatocytes. And all of those is basically, all of those bile canaliculi are collecting into the ductules, which drain into the bile duct. So the flow of blood is from outside to inside. The flow of bile is from inside to outside. The bile is going into the ducts, and the ducts are going to fuse into the common bile duct. So damaging our liver can lead to other effects. It can lead to degenerative changes, constriction of blood flow, viral hepatitis, alcoholism examples. Constricted blood flow from a clot or damaged tissue can cause portal hypertension, which is going to be basically an increase in pressure in the hepatic portal system, descending the peripheral veins, capillaries, could lead to a rupture of those vessels, could be potentially fatal. It could also force fluid into the peritoneal cavity because it changes the pressure, and if the pressure's changed, then the filtration and reabsorption are not going to occur properly, and therefore fluid can build up. That's a condition called ascites, which I referred to before. Here is, and you know what, I think I'm going to stop because I have some things I got to do. So we'll stop this video here. You can go back, review these 3 questions. Hopefully the liver lobules make sense to you. It's probably one of the trickier parts of this whole chapter, other than the digestive enzymes. So see ya.